Let's begin our lecture. And today's topic is respiratory system. It's one of the visceral systems and it provides very important functions. And today we'll talk about the structure, microscopic structure of the organs of respiratory system, and also how do those structures provide own functions. So, respiratory system includes such anatomical regions and organs as nasal cavity. It includes pharynx, which has nasopharynx, oropharynx, and laryngeal pharynx. Also, we have larynx. Here we have trachea, also bronchi, main extrapulmonary and also intrapulmonary bronchi, which together with respiratory portion of the respiratory system form lungs. So there are organs: nasal cavity, pharynx, larynx, trachea, bronchi, and lungs. And today we'll talk about their microscopic structure. So respiratory system includes different organs, and also those organs may be divided into upper respiratory tract and lower respiratory tract. Upper tract it includes nasal cavity, pharynx, and larynx. And lower respiratory tract, it includes trachea, bronchi, and lungs. This division is mainly clinical, and different specialties of medicine deal with those parts of respiratory system. For example, upper respiratory tract is treated by otorhinolaryngologists, and lower respiratory tract is treated by therapeutists and pulmonologists. So different doctors deal with different parts of respiratory tract. It's clinical division, and also we have functional division and structural, and later we'll talk about this. So the main function of respiratory system is external respiration, or it provides gas exchange. We release carbon dioxide into outer environment and we receive oxygen, and this exchange is provided by respiratory system. And according to the functions of respiratory system and features of these functions, we divide respiratory system into conducting and respiratory portions. Conducting portion, according to this name, it conducts air, and respiratory portion, it provides respiration, it provides gas exchange. So we have ways for air inside our body which conduct this air. And respiratory portion, it includes very small vesicles which have very thin wall, and those alveoli, those vesicles, they provide respiration, gas exchange. But we need also ways for air, and air should somehow get to the respiratory portion. That's why we have these two parts of respiratory system. So conducting portion, it includes nasal cavity, it includes pharynx, larynx, trachea, bronchi, bronchioles, preterminal and terminal bronchioles. So all those structures, nasal cavity, pharynx, larynx, trachea, bronchi, extra and intrapulmonary, and bronchioles, they conduct this air, and air gets to respiratory portion via all those structures. And it's a system of ducts, different ducts, which conduct air to alveoli in the lungs. And what are the functions of the conducting portion? First of all, it's conduction of the air, and also it provides this portion. It provides conditioning of the air, including warming, moistening, and removal of particular materials, different dust particles, different bacteria, something else. All those structures are removed from air, and clean, warmed, and moistened air gets to alveoli. Also, a conducting portion provides phonation, its voice generation, and also its important function of the respiratory system. Also, nasal cavity, olfactory region of the nasal cavity, it provides smell, its sensory function. And also, this conducting portion provides function of thermoregulation. Extra warm, extra heat is released from our organism via breathing. Warm air is released, and this way conducting portion provides thermoregulation. For example, when we are located in the warm places, when we feel heat, we are breathing more faster, and it's because of thermoregulative function of conducting portion. More heat is released into outer environment. And respiratory portion, it provides the main functions of respiratory system. It provides external respiration, gas exchange. We release carbon dioxide and we receive oxygen from air, from outer environment. And respiratory portion, it includes respiratory bronchioles, alveolar ducts, alveolar sacs, and alveoli, main structures, basic structures of respiratory portion. And today we'll talk about their structure, microscopic structure, and how do they provide their functions. And also some other functions of respiratory portion and respiratory system are immune system, immune function, endocrine function. Also, it provides blood deposition in the small blood vessels of lungs. Blood is accumulated. Also, regulation of blood clotting. Some factors of blood clotting are produced in the lungs. Detoxification. Some substances they undergo their final stages of metabolism in the lungs. Participation in water, mineral, and lipid metabolism. There are main functions of respiratory system and also respiratory portion of respiratory system. And let's begin from the conducting portion. And conducting portion is formed by different tubes, different tubules, and they form airways. Those ways which provide way of air from outer environment to respiratory portion. Airways, they include nasal cavity, pharynx, larynx, trachea, bronchi, terminal bronchioles, and all those structures, they have 4 layers of the wall. They are hollow structures, they have cavity, and they have wall, which surrounds this cavity, and this wall, usually it has 4 membranes. Those membranes are mucosa, submucosa, fibrocartilaginous layer, and adventitia. And let's look here. It's mucosa, it's submucosa, fibrocartilaginous layer, and adventitia. 4 basic layers of the wall of airways, of conducting portion of respiratory system. Here we can see mucosa, it's submucosa, fibrocartilaginous layer, and adventitia. And I think you remember about histology of digestive system, especially digestive tract, and you remember that there are layers of the wall of digestive tract. And here we can find at least 3 layers which are similar to those in digestive system. It's mucosa, submucosa, and some organs of digestive system, they have adventitia. But here we have fibrocartilaginous layer, which is composed of cartilage and dense connective tissue, and it's a specific feature of respiratory system. This layer, it maintains the shape of airways. They aren't collapsed, and their shape is maintained by the cartilage, which is located in this layer. And let's talk about all those layers. So the innermost layer is mucosa. Next it's submucosa. Here we can see. Next it's fibrocartilaginous layer. And the outermost layer is adventitia. 4 basic layers of the wall of respiratory tract. So the innermost layer is mucosa. And mucosa, it is little bit similar to mucosa in digestive system, and it has 3 own sublayers. It's epithelium, it's lamina propria, next layer it's Vascularis Mucosa, and there are 3 layers which are similar to the layers in the digestive tube. Also there are epithelia, lamina propria, and Vascularis Mucosa. And specific feature of the airways, it's epithelium. Here we can see this epithelium, and we can see that it's different. It is not the same as in the digestive system. So let's classify this epithelium. Epithelium of respiratory mucosa is pseudostratified columnar ciliated. Here we can see epithelium. Nuclei are located on the different levels, so it has appearance of stratified epithelium, but all cells they have contact to the basement membrane, and that's why we classify this epithelium as simple. And because of appearance of this epithelium, we classify them, we call them this epithelium pseudostratified, because it reminds us of stratified epithelium. This epithelium is columnar, and cells they have cilia, and this type of epithelium is called ciliated. And there are main cells of the epithelium of respiratory mucosa. So we have here ciliated cells, we have goblet cells, basal cells, also there are brush cells or cells with microvilli, endocrine cells, and in lower parts of respiratory tract, in bronchioles, we have Clara cells also. So here we can see ciliated cells, cells with cilia. Between them we have secretory cells. They are goblet cells. Also we have brush cells or cells with microvilli. Also we have intercalated and basal cells. Later we'll talk about them. And also we have endocrine cells near the basal pole of the ciliated cells, near to the basement membrane. So here we can see main cells of the respiratory epithelium of pseudostratified ciliated epithelium, which lines organs of respiratory tract. And here we can see this epithelium. It's drawing of histological slide, and we can see here goblet cells, ciliated cells, also basal cells are present here. They are located near the basement membrane. They are located deeper. And the most important cells of those plenty of cells in the respiratory epithelium are two types. There are ciliated and goblet cells. They are the most numerous and the most important cells. So please remember those types of cells, ciliated and goblet cells. Ciliated cells, they have cilia. Here we can see movable projections of the apical pole of the cell. And goblet cells, they have inclusions of mucus, so they are secretory cells, and they provide secretory function. They produce mucus. And those cells, they are related to each other. Those relations, they result in the common function. And this common function is mucociliary clearance or mucociliary transport. Here we can see ciliated cells, they have cilia on the apical surface, and goblet cells, they secrete mucus that covers the surface of the epithelium. So let's look how do they work together. Here we can see cilia of ciliated cells, and they are covered with layer of mucus. Here we can see this mucus layer, and different dust particles are attached to this mucus. And this mucus is moved by the cilia of ciliated cells. They move a layer of mucus and remove different foreign particles. Here we can see cilia and its mucus layer. Air comes here, and different dust particles are attached to the mucus, and cilia they move and remove this layer of mucus. Here we can see goblet cells and ciliated cells. They are the main two types of this epithelium. And here we can see production of mucus. Special staining methods, they can reveal this mucus in the goblet cells. So those blue points, they show us they mean inclusions of mucus inside the goblet cells. And they release this mucus via apical pole, via exocytosis. Here we can see, and this mucus is dissolved, and it forms parietal layer of mucus which covers the cilia, and cilia are covered with layer of mucus. And when breathing, different particles are coming here and they are attached to the surface of the mucus. For example, dust particles, some bacteria, something else. Air isn't very clean, so this air it contains something foreign. And all those particles they are attached to the layer of mucus. And cilia, they are movable and they make movements and they move this layer of mucus together with foreign particles. And here we can see movements of cilia and they move in the distal direction from the lungs to outer environment. Those layer of mucus and particles of dust and different foreign particles. And here you can see how cilia of ciliated cells are moving layer of mucus with foreign particles. So this cooperation of goblet cells and ciliated cells is called mucociliary clearance or mucociliary transport. So main cells are ciliated and goblet cells. But also we have their basal cells, brush cells, endocrine cells, Clara cells, and let's look what are those cells. First of all, there are basal cells, and also here we can see basal cells. They are short, small cells located near the basal membrane. And also we have intercalated cells. They are taller than basal cells, and they are located between tall ciliated cells. Here we can see basal cell and intercalated cell. And presence of those cells, it results in the feature that nuclei of the cells, they are located on the different levels. And they form some rows. That's why we have we can see appearance of this epithelium which is similar to appearance of stratified epithelium. So what are those basal and intercalated cells? They are young cells which provide regeneration and basal cells. They are like stem cells, some kind of stem cells, and they are growing and they become to be intercalated cells which may become to be any of superficial cells, ciliated, goblet, or brush cells. So let's look how is it provided. Here we can see ciliated cells, and between them near the basal pole we can see small basal cells. And this basal cell, let's look at this cell, it grows. And it grows between the ciliated cells and it becomes to be intercalated cell. Here we can see intercalated cell, it's result of growth of the basal cell. And what happens next? After that, this cell continues growing, it keeps growing and it pushes the ciliated cells and is located between them. And for example, in this case, this cell becomes to be ciliated, another ciliated cell, cilia appear. And here we can see how basal cell provides regeneration of this epithelium. It replaces ciliated cell. It becomes to be ciliated cell. So it's the stem cell which can become to be any of those kinds of cells. And it's how epithelium regeneration is provided in the respiratory system. Also, we have brush cells. There, they are described as cells with sensory function. They have microvilli on their surface. And we have also endocrine cells. They are similar to those which are present in the digestive tract. And they produce some hormones which regulate functions of the neighboring tissues and structures. And also in lower respiratory tract, in bronchioles, we have special Clara cells. They also are secretory cells as well as goblet cells, but they produce surfactant, special substance which reduces surface tension in lower respiratory tract, and we'll talk about this Clara cells functions. So they are located deeper near the they are located in the bronchioles and near the respiratory portion. And under epithelium, we can see layer of loose fibrous connective tissue, and this layer is called lamina propria. Lamina propria is composed of loose fibrous connective tissue with blood vessels, with nerves. These connective tissues, they always follow epithelia. So all epithelia under their basal membrane, they have thin layer of loose fibrous connective tissue. So it's essential layer which provides nutrition of the epithelium. It maintains the epithelium. Also, some immune cells are present in lamina propria, and this layer is similar to the same layer in the digestive system. And under lamina propria, we have Vascularis Mucosa. Here we can see this Vascularis Mucosa. It's composed of smooth muscular tissue, a thin layer of smooth myocytes. And this Vascularis Mucosa, it can change relief of mucosa, contractions of the smooth myocytes, and this Vascularis Mucosa is developed in different degree in different regions of respiratory system. Vascularis Mucosa is well developed in lower parts of Bronchial Tree, in small bronchi, and later we'll see what is the clinical significance of this Vascularis Mucosa, because it's very important. And under mucosa, we have next layer, it's submucosa. Submucosa also is composed of loose fibrous connective tissue. And in some places, Vascularis mucosa isn't well developed, and we cannot see clear border between lamina propria of mucosa and submucosa, because both layers are composed of loose fibrous connective tissue. It contains mucus glands, it contains blood vessels, nerves, and also lymphoid tissue. Here we can see glands which produce mucus, which is released in the lumen of the respiratory tract. It covers epithelium, so it also helps in the mucociliary clearance. Also, there are blood vessels which provide blood supply of submucosa. Also, nerves are present here, which especially innervate Vascularis mucosa and in other structures. And lymphoid tissue is present here, which provides immune function. And we have bronchus-associated lymphoid tissue, which is represented by lymphoid follicles, which are present in submucosa mainly, but also they may be present in mucosa of respiratory tract. And under submucosa, we have the next layer. This layer is called Fibrocartilaginous layer. Why do we call this layer fibrocartilaginous? It is composed of cartilage. Mainly, there are hyaline cartilages. They form different particles, different structures. For example, cartilaginous islets, cartilaginous plates, or rings, or different cartilaginous structures. And they are surrounded with dense fibrous connective tissue, which maintains and connects those cartilages. So we have fibrous bases with inclusions of cartilage, or this layer may be completely formed by the cartilage. So here we can see a particle of the hyaline cartilage, and it's surrounded with dense fibrous connective tissue. And this layer may be composed of two types of cartilaginous tissue. It may be hyaline cartilage, mainly hyaline, but in some places we have elastic cartilage. For example, in the larynx, some cartilages are elastic. So here we have fibrocartilaginous layer. And main function of this layer is to maintain open lumen of respiratory tract. In the digestive system, here we have muscularis externa, which provides peristalsis. Food is denser than air, and we need peristalsis to move this food through the digestive tract. But in respiratory system, air is located in the lumen, and lumen should be kept open to provide breathing, and we should prevent collapsing of the respiratory tract. And this requires um, maintaining structures in the wall of respiratory tract. And we have here cartilages which maintain the wall of respiratory tract, and they form this wall, so it's a basis of the respiratory tract, of the organs of respiratory tract. So cartilages, they are like skeleton of respiratory airways. And here we can see cartilages, and they maintain lumen of the bronchus or of the trachea, the larynx. They keep it open to provide respiration, breathing, and air can pass through the respiratory tract. And also we have the outermost layer, it's Adventitia. Here we can see this layer. It's composed of loose fibrous connective tissue which covers bronchus or trachea or another organ outside, and it contains blood vessels and nerves. And Adventitia is similar to Adventitia in other organs, for example, Adventitia in the digestive system, in digestive tract. And important note here, in digestive tract we have two kinds of outer membrane. It may be serosa or adventitia. Serosa because some intestines, they are covered with peritoneum. In respiratory system we haven't serosa covering of the bronchi of trachea, so only adventitia is present here. Only adventitia is the outer layer of the respiratory tract. And here we can see a drawing of histological slide. It's trachea, and here we can see mucosa. Here we can see mucosa. Next, submucosa with glands. Next, it's fibrocartilaginous layer. It's cartilage and it's fibrous membrane. And the outer layer, it's adventitia. Here we can see it's mucosa, epithelium, and lamina propria, and Vascularis Mucosa isn't well developed. Next, it's submucosa with glands. Here, fibrocartilaginous layer is present. It's cartilage, hyaline cartilage, and its fibrous membrane. And this is adventitia with blood vessels, and also adipose tissue may be present here. And here we can see trachea. Also we can see here it's mucosa. Under the mucosa we can see a thin layer of submucosa. We can identify it by the presence of glands. Next, it's fibrocartilaginous layer, it's cartilage, and the outermost layer is adventitia. And let's compare digestive system, which you already have studied, and respiratory system. So both they have mucosa, and mucosa is composed of epithelium, but in respiratory system epithelium is specific. Next, it's lamina propria. They have almost the same structure. Vascularis mucosa. Next, it's submucosa. They are a little bit similar, but in the respiratory system, it mainly contains glands. In the digestive system, not all organs they contain glands in submucosa. Next, vascularis externa is present in the digestive system. In respiratory, we have fibrocartilaginous layer, and the outer membrane in digestive system may be serosa or adventitia. In respiratory system, always it's adventitia. And here we can compare. So main difference is in the vascularis externa and fibrocartilaginous layer, which results in the different features of the structure of those organs. And let's talk about different organs of respiratory system. So you already know the layers of the respiratory tract, and now let's look at the regional features and differences. So, and we will begin from upper airways, and we'll begin from the nasal cavity. Nasal cavity, it has two anatomical regions. It has vestibule, and it has nasal cavity proper, which has olfactory and respiratory region. Vestibule, it has two layers. It's skin, which is continuation of the skin of the face of the outer surface of nose. It enters in the vestibule of nose, and it contains here hairs, which also filtrate some dust particles from outer environment. And basis of this region is cartilage, and outer cartilages of nose, they are mainly elastic, and you can feel this elasticity. You can touch your nose, and you can feel that there is elastic cartilage. But septum of nose is composed of hyaline cartilage. It's much more denser. Um, and there are layers in the next region, in the respiratory region and olfactory region. We have here mucosa instead of skin. So here in respiratory region and olfactory region, we have mucosa, its epithelium and lamina propria. And in some textbooks you can find that there is submucosa, and some textbooks you can find that there is only lamina propria. What is the reason of this? Here we can see that Vascularis Mucosa is that well developed, and we cannot see a clear border between lamina propria and submucosa. And we can see that appearance that lamina propria is continued by submucosa. So some scientists describe this layer as mucosa, lamina propria of mucosa, and some scientists they describe submucosa as a distinct layer. So it will not be a mistake to classify in both ways. So mucosa is epithelium, lamina propria is loose fibrous connective tissue. Deeper we have submucosa, which is composed of loose fibrous connective tissue, and it contains mucous glands which produce mucus, which covers epithelium of nasal cavity. And deeper we have bone or cartilage, according to the anatomical localization of the part of nasal cavity, depending on region. It may be bone, which forms nasal cavity, or cartilage, mainly hyaline, which forms part of the septum, nasal septum, and its basis, which underlies mucosa and submucosa of nasal cavity. And here we can see epithelium, covering epithelium, it's lamina propria, and deeper we have glands here, and under the glands, under lamina propria and possibly submucosa, we can see here bone, or it may be cartilage here. And here we can see frontal section of the nasal cavity. We can see here nasal septum, its bony part, its cartilaginous part. Here it's hyaline cartilage, and there are concha of the nasal cavity, and also structures they are covered with nasal mucosa, and we can see it. And in lamina propria, in submucosa, there are well-developed blood vessels which provide warming of air, and also result of this good development of blood vessels is bleeding, intensive bleeding after trauma of nose, because blood vessels are well developed in mucosa and submucosa of the nasal cavity. And there are two regions, it's respiratory and olfactory region, and they have differences in their covering epithelia. In respiratory region, we have pseudostratified ciliated epithelium, those epithelium which we already know, with goblet cells which produce mucus, and this epithelium provides protective function, it provides mucociliary clearance. And in the olfactory region here, we have special olfactory epithelium which provides sensory function. It provides sense of smell. Here we can see special olfactory cells. And let's look at this epithelium. So olfactory epithelium, it has main cells. They are olfactory cells. They are modified neurons. They have cell bodies. they have modified dendrite with cilia-like projections. Here we can see, and also these cells they have modified axons. So those cilia they receive information, conduct this information to the olfactory cells, and they conduct this information to CNS. Between olfactory cells, we have supporting cells or maintaining cells, and also we have basal cells here. They provide regeneration. And under the olfactory epithelium, we have basement membrane and lamina propria. And olfactory epithelium also is covered with layer of mucus. And when we are breathing, different smelling particles, molecules are coming here. They are dissolved in the layer of mucus here, and they irritate, they activate cilia on different olfactory neurons. They are activated and they send information to CNS. So here we can see a cell, and it uptakes information, generates nerve impulse, and this nerve impulse runs through the axon to CNS. And here we can see olfactory epithelium and appearance of this olfactory epithelium under microscope. There are olfactory cells and cilia. Their cilia are present on the surface of olfactory epithelium. Here we can see connective tissue with blood vessels. It's lamina propria and submucosa, if we describe them as distinct layers. And here we have hyaline cartilage, or it may be bone, according to the region. And axons, modified axons of olfactory cells, they form olfactory nerve. Axons, their bundles, they pass through the lamina cribrosa, and they are going to the olfactory bulb, and it sends impulses to the CNS. So it's formation of the olfactory nerve. And now we are going to the next organ of the respiratory system. It's pharynx. It has three anatomical regions. It's nasopharynx, oropharynx, and laryngopharynx. And pharynx, it is organ which is part of respiratory and digestive systems, and it has features characteristic for both systems. But mainly, it's similar to the esophagus. It has mucosa, submucosa, vascularis, and adventitia. So it's more similar to the organs of digestive system. Mucosa, submucosa, muscularis, and adventitia. Muscularis of pharynx is formed by skeletal muscles which are voluntary and they participate in the swallowing, and we can control them with our mind. And there are differences in different parts of the pharynx. In oropharynx and laryngopharynx, we have stratified squamous and keratinized epithelium, which is present also in the esophagus, which is continuation of the pharynx here. So this epithelium is continued in the esophagus. But in nasopharynx, in this region, we have epithelium pseudostratified ciliated, which is present in the nasal cavity and respiratory system. So in the nasopharynx, we have this type of epithelium. In oro and laryngopharynx, we have this type, stratified squamous non-keratinized. And we are going to the larynx. So pharynx is opened to the larynx, and larynx it's an organ of only of respiratory system. So it has typical structure of organs of respiratory tract. And larynx, it has four layers. It's mucosa, submucosa, fibrocartilaginous layer, and adventitia. Typical structure. And what are the features of the larynx? So mucosa is lined with pseudostratified ciliated epithelium, but one exception is vocal folds. Here we can see here it's vocal fold. There are false and true vocal cords, upper and lower, and true vocal folds are covered with stratified squamous non-keratinized epithelium. This epithelium, it hasn't cilia, it's stratified epithelium because these mucosal folds, and they form vocal folds, they participate in the vocation, voice generation, and cilia aren't needed here, and because ciliary clearance also, we need here more strong epithelium on the vocal folds. But false vocal folds, they are covered with typical pseudostratified epithelium. And one more difference that in true vocal folds we have vocal muscle, it's skeletal voluntary muscle, and in false we haven't this muscle, it's only fold of mucosa and submucosa with glands also. Fibrocartilaginous layer, it's composed of hyaline cartilaginous tissue mainly, but there are some exceptions. Some cartilages, cuniculate cartilages epiglottis, they are composed of elastic cartilaginous tissue, so this tissue is present here instead of hyaline. So some cartilages of the larynx, they have elastic cartilage, not hyaline, but mainly there are hyaline cartilages. And their shape, it has strict anatomical parts, so you study them at anatomy, and you should know that in histological level they are composed of hyaline cartilaginous tissue, and some cartilages are elastic. Next organ of respiratory system, of respiratory tract, it's trachea. And trachea, it has four membranes, typical membranes for the respiratory tract. It's mucosa, submucosa, fibrocartilaginous layer, and adventitia. And now let's talk about features. What are the features of trachea? So first of all, mucosa is lined by pseudostratified ciliated epithelium. So it's a typical epithelium. Here we can see its appearance. In submucosa, submucosa is composed of loose connective tissue, and there are mucus and serous glands. And in submucosa of trachea, they are well developed, better than in the following parts of respiratory tract. So here we can see numerous glands. It's epithelium, it's lamina propria, and in submucosa we can see glands. They may be mucus, they may be serous, which are very similar to the salivary glands, to small salivary glands. And these glands, they produce mucus, they produce serous secret, which covers surface of respiratory mucosa. Next layer, it's fibrocartilaginous layer, and feature of fibrocartilaginous layer that it's composed of C-shaped hyaline cartilages, which are connected by fibrous membrane. Here we can see C-shaped cartilages, and posterior wall of trachea isn't cartilaginous. It's soft. Posterior wall of trachea is soft. Anterior and lateral walls of trachea, they are hard because they are composed by cartilage, and they maintain the shape of trachea, but posterior wall is soft. And here we can see this cartilage, and there is a fibromuscular membrane which connects the ends of C-shaped cartilage. So what is the reason of this? Let's look at the anatomical localization of trachea. Trachea is located in the front of the esophagus, and esophagus is located near the trachea, and on the cross section, on transverse section, we can see here trachea, its lumen of trachea, and its esophagus. And when we are eating something, when swallowing, food passes through esophagus, and esophagus in those cases it needs more place to conduct this food, and the lumen of esophagus is dilated, and food passes through this lumen. But esophagus needs more place, and posterior wall of trachea is flexible here. This wall is flexible, so esophagus takes some place which is filled, which is taken by trachea, And this flexibility of the posterior wall of trachea, it helps to provide swallowing, and food moves this posterior wall of trachea. But please, students, notice that food doesn't pass through the lumen of trachea, it passes through the lumen of esophagus. But esophagus takes some place, some volume from trachea, and this flexible posterior wall of trachea, it admits swallowing and passage of food through the esophagus. So we need to have this flexible posterior wall of trachea to provide normal swallowing. Here we can see drawing of histological side of trachea. You have seen this slide. So it's epithelium of mucosa, it's lamina propria of mucosa, it's submucosa here, and we can find here glands. It's fibrocartilaginous layer, it's cartilage, and it's fibrous membrane. And this fibrous membrane also can contain Mucus, which provides some contractions of this membrane. And the outer layer, it's Adventitia, which contains blood vessels. Here we can see its epithelium, its lamina propria, submucosa. It has well-developed glands, and look here, there are serous glands, mucus glands, and even mixed secretory units with serous demilums are present here. And it's hyaline cartilage. It's part of the C-shaped ring, hemi-ring of trachea, and its fibrous membrane, and its adventitia. And its histological microphotograph of trachea, its epithelium of mucosa and lamina propria. Deeper we have submucosa, its cartilage, and here we have adventitia. And also here we can find well-developed C-shaped cartilage of trachea and its posterior wall. Here we have posterior flexible membrane of trachea. And now we are going to next organ of respiratory system. We have lungs and we have bronchi. So first of all, we will talk about bronchi, which form bronchial tree inside lungs, and also we have extrapulmonary bronchi, right and left principal bronchi. So let's talk about their structure. So lungs, they have two lobes in the left lung and 3 lobes in the right lung. Here we can see upper, medial, and lower lobes. And lobes are divided into segments, and in the right lung we have 10, and in the left lung, by different textbooks, we have 10 or 9 segments here. And all those parts of lungs, they receive air from bronchi, and bronchi, they are branched in the lungs according to these divisions of the lobes. So here we have also conductive portion. It includes bronchi, preterminal and terminal bronchioles, which form bronchial tree. And also we have respiratory portion, which includes respiratory bronchioles, alveolar ducts, sacs, and alveoli. So we'll begin from conductive portion. It's the bronchial tree. We have trachea here, and it's divided into right and left principal bronchi, which are divided into lobar bronchi. Lobar are divided into segmental, and segmental they undergo further divisions. And here we can find that bronchi usually are divided into 2 branches, and this pattern of divisions we can call dichotomous division, division into 2. And here we can see dichotomic divisions of bronchi of different generations. Here we can see principal bronchus, which ventilates, which provides, which supports air to entire lung. It's principal bronchus. After that, it's divided into lobar bronchi. In the left lung, we have two lobar bronchi for upper and for lower lobes, and in right we have three lobar bronchi for upper, middle, and lower lobes. And after that, lobar bronchi, they are divided into branches which support air for segments, segmental bronchi. And after that, they form about 20 generations of divisions. So they are divided into two secondary bronchi, and each secondary is divided into tertiary and following generations, and about 20 generations of bronchi we have in our lungs. And here we can see different divisions of bronchi, and we have very, very small bronchi of small generations, and all those bronchi, they form bronchial tree. It's conductive portion which is located inside the lungs. So we have lobar and segmental bronchi and bronchi of following generations till pre-terminal and terminal bronchioles, and all those branches we call bronchial tree. And here we can see that there are right and left principal bronchi. They form lobar bronchi. Lobar are divided into segmental bronchi, and there are following generations. And the last are preterminal bronchioles and terminal bronchioles. They are the last branches of the conductive portion. Here we can see generations. And they provide ventilation, or they provide support of air for different regions of lung. So right and left principal bronchi, they carry air to right and left lobes. They support air ventilation for the entire lobes, so right and left lungs, and they provide their ventilations. Lobar bronchi, they support air to the lobes, three lobes in right lung and two lobes in the left lung. Segmental bronchi, they carry air to the segments of the lungs, 10 in right lung and 10 or 9 in the left lung. And after that, we have units of the lungs. Pre-terminal bronchiole, it supports air to the region of lung which is called lobule, and terminal bronchiole, it supports air to the part of the lung which is called acinus. And let's look at the structure, first of all, at the structure of the bronchi. So bronchial tree and bronchi, they have typical structure. They have mucosa, submucosa, fibrocartilaginous layer, and adventitia for typical layers. And let's look at the differences. So here we can see it's mucosa, and it's Vascularis Mucosa. Deeper we have submucosa with well-developed glands. We can see them here. There are secretory units of glands. Here we can see cartilaginous structures. It's fibrocartilaginous layer, and the outer layer it's adventitia, which is continued into the parenchyma of lung. And according to the diameter of bronchi, we can divide all them into three groups. We can describe large, medium, and small bronchi. Large are principal and lobar bronchi. Medium bronchi, they are segmental and subsegmental bronchi, which are branches of segmental bronchi. And small bronchi are those which are formed by following divisions. They are interlobular bronchi till bronchioles. So here we can see three groups of bronchi: large, medium, and small. And we can see here that all those parts, all those groups of bronchi, they have differences in the structure of their wall. And let's look at these differences. So let's follow all layers of the bronchial wall, and let's find the differences in the structure of the large, medium, and small bronchi. So here we can see the innermost layer, it's epithelium of mucosa, and we can find here gradual decrease in thickness of epithelium and number of rows. Here we can see tall cells and numerous rows in large bronchi. In medium bronchi, epithelium is thinner and cells are shorter, and in small bronchi, epithelium is the thinnest, and it goes even till cuboid type. So let's look at the appearance of this epithelium. In the large bronchi, we have tall cells. After that, cells in the following generation they become to be shorter, and epithelium is thinner. And in the small bronchi, we can see thin epithelium, which may be even cuboid. Under epithelium, we have lamina propria, loose fibrous connective tissue, and we also can see gradual decrease in thickness. Here you can see the thinnest lamina propria is present in the smallest bronchi, and I think it's easy. But the next layer, it is specific layer, it's Vascularis Mucosa, and please look at the thickness and number of layers of smooth myocytes. The thinnest Vascularis Mucosa is present in the large bronchi, but in the small bronchi it's well developed. So here we can see gradual increase in thickness of Vascularis Mucosa. If you remember trachea, in trachea this Vascularis Mucosa is almost absent. It's not well developed in trachea. But in the bronchi, we can see here gradual increase in thickness, and the best development of Vascularis Mucosa is present in small bronchi. And I told you before that this results in very big clinical significance. And you should know that small bronchi, they are able for bronchospasm, for contraction because of presence of well-developed Vascularis Mucosa. Contractions of Vascularis Mucosa, they smaller, they make lumen of bronchus smaller, and it is called bronchospasm. And it occurs in some bronchial diseases, in bronchitis, and especially in asthma, bronchial asthma. And you should know that different myorelaxants should be given to your patient which have target cells in Vascularis Mucosa to decrease this bronchospasm. There are special medicines which can reduce this spasm of these muscles, and there are target structures, target cells in the Vascularis Mucosa. So big bronchi, they aren't able for bronchospasm because they haven't well developed Vascularis Mucosa. But small bronchi may be contracted, and you should know about that. Submucosa, next layer. Submucosa is decreased in thickness. Here you can see gradual decrease in thickness of submucosa. And also number of glands also decreases. And small bronchi, they haven't a lot of glands. They are almost absent there. Next layer, it's fibrocartilaginous layer. It also decreased in thickness, and also we can see here some qualitative changes in fibrocartilaginous layer. So here you can see that in large bronchi we have cartilaginous plates and cartilaginous rings. In medium bronchi we have cartilaginous plates and islets, and small cartilaginous islets are present in small bronchi. Here we can see them. So here we can see big plates, small plates or islets, and small cartilaginous islets are present in small bronchi. So plates, islets, or small plates and small cartilaginous islets are present in small bronchi. There are qualitative changes of fibrocartilaginous layer of bronchi. Next layer is Adventitia. Here we can see it. It's the outermost layer, and also we can see here gradual decrease in thickness. So Adventitia is thinner in the small bronchi. So here we can see that all layers they have gradual decrease in their thickness, except Muscularis mucosa. Muscularis mucosa increases in thickness in small bronchi. So it's one exception, but this exception it has very big clinical significance. And let's look at the histological slides of bronchi. Here we can see large bronchus and its mucosa, its respiratory epithelium. Under the epithelium we have lamina propria, and deeper we have smooth myocytes here. Its submucosa, it's present here, which has well-developed glands. Its cartilage, its hyaline cartilage, and here we have adventitia. Its medium bronchus, it has cartilaginous islets. Its mucosa lined with epithelium. Its muscularis mucosa, we can find it here. It is a layer of glands in submucosa, fibrocartilaginous layer, and adventitia, which is continued into the tissue of lungs. And here we can see small bronchus. Here we can see epithelium of mucosa, its lamina propria, vascularis mucosa, and here we can see islet of cartilage and adventitia, which is continued into the surrounding tissues. And please, students, look here. You should know that contractions of bronchi, they are provided by muscularis mucosa, small sublayer of mucosa, but not by muscularis externa. Muscular layer at muscularis externa is absent in bronchi and bronchial tree. Muscularis mucosa is present, and this layer is able for contractions, but not the muscularis externa, which is absent in the respiratory system. There are bronchi of different generations. They provide ventilation of different parts of the lungs, different regions of the lungs. And after all the divisions, bronchi, they go to bronchioles. We have pre-terminal and terminal bronchioles, which are part of the conductive portion of respiratory system. And bronchi, at some moment, they become to be bronchioles. So why do we call some structures bronchioles and some structures are bronchi? What is the difference between bronchus and bronchiole? So relative quantitative changes in bronchi, which we already know, at some moment they lead to qualitative changes in the structure and physiology of the bronchi, and at some moment the smallest bronchi are called bronchioles because qualitative changes occur in the smallest bronchi, which are at some moment called bronchioles. And here we can see structure layers of the bronchus. Here we can see mucosa, submucosa, fibrocartilaginous layer, and adventitia. In bronchioles, what do we see here? What are the changes? Also, we can distinguish four layers. It's mucosa with epithelium, lamina propria, muscularis mucosa. Next, submucosa. Submucosa, it lost all glands, so it's empty. Fibrocartilaginous layer. In fibrocartilaginous layer, we cannot find any cartilage, so cartilage, they disappear. So glands disappear, cartilage disappear. And let's look at the epithelium. In epithelium of the bronchus, we can see goblet cells, and in the epithelium of the bronchiole, we cannot find goblet cells. We can find here Clara cells. They replace goblet cells. They are present here instead of goblet cells. And let's look at those features and why do they occur. Let's discuss this. So first of all, cartilage disappear. It's first qualitative change, and it results in the soft wall of the bronchiole. So wall of the bronchiole isn't supported by the cartilage, and cartilage doesn't support this wall, and it may cause collapsing of bronchioles. They may be collapsed, and to prevent this collapsing, There is special layer which lines the bronchioles. It's layer of surfactant. Here we can see bronchial epithelium and it's covered with layer of mucus, which is produced by goblet cells. But in bronchioles we have layer of surfactant, special lipid substance which covers surface of the epithelium and it prevents collapsing. It's present in all parts of respiratory portion. It lines inner surface of the alveoli, and in bronchioles it also prevents their collapsing. So Clara cells, they produce surfactant. They have this shape and they have inclusions of surfactant. They are secretory cells, and surfactant prevents collapsing. In bronchi, we have goblet cells which produce mucus, but bronchi they are supported by the cartilage which maintains their shape. But in bronchioles we haven't this basis, we haven't this support. So in bronchioles, mucus uh, may cause collapse of the bronchioles, and that's why we have in another lining it's surfactant which prevents this collapsing. And also that's why glands disappear because they produce mucus. All mucus producing components of the wall they disappear. Goblet cells and mucus glands, because mucus shouldn't be present here because mucus may cause collapsing of bronchioles. Another substance is produced here. It's surfactant which covers the surface of the bronchiole. So changes in bronchioles are qualitative and they are different structures. They aren't the smallest bronchi, they are another structures which continue bronchi. They have another name, and here we can see difference between the bronchus and bronchiole. So in bronchus we have goblet cells. In bronchiole, goblet cells are absent. Clara cells are absent in bronchi, but they are present in bronchioles. Glands and cartilages are present in bronchi and absent in the bronchioles. So here we can see main changes which occur when bronchus becomes to be bronchiole. And we have two generations usually of bronchioles in the conductive portion. There are preterminal bronchioles and terminal bronchioles. So the smallest bronchi, small bronchi, they are divided into the branches which are called preterminal bronchioles. Here we can see preterminal or terminal bronchiole. It has well-developed muscular layer. It's Vascularis Mucosa. We can see here surrounding Adventitia, but cartilage is absent here. And region of lung, which is ventilated by preterminal bronchiole, is called lung lobule. It's structural unit of the lung. It's region of lung, pyramidal shaped region, which receives air from small bronchiole, which is called preterminal bronchiole, and it's called lung lobule. And here we can see preterminal bronchiole. It has well developed Vascularis Mucosa, a layer of smooth myocytes, but cartilage is absent here. It's mucosal folds and it's the lumen. Preterminal bronchioles, they are divided into two terminal bronchioles. Here we can see this terminal bronchiole. And terminal bronchiole, it has thinner wall. In the wall we have epithelium and also lamina propria, thin layer of loose fibrous connective tissue. But muscularis mucosa almost disappears, so epithelium and connective tissue bases mainly are present in the terminal bronchiole. And the region which is ventilated by the terminal bronchiole, region of the lung, is called pulmonary echinus. Pulmonary echinus is region of lung, unit of lung, which receives air from the terminal bronchiole. So preterminal bronchiole ventilates lobule, and terminal bronchiole it ventilates echinus. Terminal bronchioles, they are the branches of the preterminal bronchioles. And here we can see terminal bronchiole. is divided into the following branches, and we'll talk about them. And all this region, which is ventilated by the terminal bronchiole, is called lung acids. So we have discussed conductive portion, which conducts the air inside our respiratory system, and at some moment air gets to the structures which provide gas exchange, and we call this respiratory portion of the respiratory system. So let's go to the structure of respiratory portion. And respiratory portion, it includes such structures which are called alveoli. They have very thin wall. They are lined with simple squamous epithelium, which provides this gas exchange between air and blood. And here we can see numerous alveoli in this region, in the respiratory portion, lungs tissue. It has sponge-like structure. Here we can see it. And there are numerous alveoli which provide gas exchange between air and blood. And here we can see alveolus, it's a capillary, and blood is circulating in the capillaries. Lungs, they receive venous blood from the heart, from the pulmonary artery, its small route of circulation, and blood comes here, it circulates in the capillaries, and it releases carbon dioxide in the air, and it receives oxygen from the air. So we can see here gas exchange which is provided via this wall. So it's respiration or gas exchange which is provided by the respiratory portion. And respiratory portion includes alveoli, the main structures, and alveoli also they form different structures, and they may be present in the inside in other structures. So we have here respiratory bronchioles, we have alveolar ducts and alveolar sacs, and all those structures they are composed of alveoli. And here we can see terminal bronchiole. We have respiratory bronchioles. Terminal bronchiole is divided into two respiratory bronchioles, and respiratory bronchioles here we can see that inside their wall there are some bubbles, and they are alveoli. Also there are alveolar ducts and alveolar sacs, and all those structures they are formed by alveoli. So let's look at this structure. Terminal bronchioles, they are divided into respiratory bronchioles, and respiratory bronchioles, they ventilate respiratory bronchiolar unit, and respiratory portion begins. So all these structures, they belong to the conductive portion, and now we are going to the respiratory portion. So what is the difference, first of all, between conductive and respiratory portion? Here we can see conductive portion, it's terminal bronchiole. It has continuous ciliated epithelium, and this region is close to cuboid. And at some moment in the wall of the bronchiole, some bubble-like structures appear, and they are called alveoli. So here we can see cuboid ciliated epithelium, and it's interrupted by simple squamous, and again cuboidal ciliated, again simple squamous. So alveoli, they are inserted in the wall of the terminal bronchiole, and this bronchiole we call respiratory bronchiole, which provides respiration, which provides gas exchange, because alveoli appear here. In the terminal bronchiole, gas exchange is impossible because epithelium is too thick for the gas exchange, but in respiratory bronchiole, special regions for gas exchange appear. That's why we classify respiratory bronchiole as a part of the respiratory portion. So here we can see border between conductive and respiratory portion. Respiration gas exchange is possible here. After that, respiratory bronchiole is continued by special duct, which is completely composed of alveoli. In respiratory bronchiole, we had islets of ciliated epithelium, but here it completely disappears, and alveolar duct is completely formed by alveoli. And the end points of the respiratory portion are the alveolar sacs which are composed of numerous alveoli. Numerous those bubbles, vesicles, they form alveolar sacs. And let's look how it's formed inside the lung. So here we can see it's terminal bronchiole. It's composed by ciliated epithelium. It's not permeable for gases, so we will show those unpermeable regions as a pink line, and simple squamous epithelium of alveoli is shown as blue lines. So it's terminal bronchiole. It's still conductive portion, and it's divided into two branches. It's dichotomic pattern of divisions. Two branches are present here, and those two branches are respiratory bronchioles. They have inclusions in their wall. They have alveoli. So two respiratory bronchioles, they are primary or respiratory bronchioles of first order. And the alveoli take about 1/3 of the wall of the area of the wall of respiratory bronchiole. After that, they are divided into two secondary respiratory bronchioles. Here we can see next branches, next generation. It's secondary respiratory bronchiole. And secondary respiratory bronchiole is divided into two tertiary respiratory bronchioles. So primary, secondary, and tertiary, three generations are present here. In the first generation, in primary respiratory bronchioles, alveoli, they take about 1/3 of the wall of respiratory bronchiole. In secondary, about 2/3, and in the tertiary respiratory bronchioles, up to 3/4, up to entire area of the wall, but small islets of epithelium, ciliated epithelium, are still present here. And tertiary respiratory bronchiole is divided into two branches, into two alveolar ducts. Here we can see tertiary respiratory bronchiole, and it's opened into two alveolar ducts. And each alveolar duct is opened into two alveolar sacs. So divisions into two following branches are saved. And here we can see the end points of the respiratory portion of respiratory system. They are alveolar sacs. And here we can see all those structures which contain alveoli. So gas exchange takes place in all those structures. And these branches of this bronchial tree formed by terminal bronchiole are called pulmonary acids. So here we can see terminal bronchiole is divided into two primary respiratory bronchioles. There are islets of ciliated epithelium and alveoli inserted in the wall of respiratory bronchiole. After that, primary, secondary, and tertiary respiratory bronchioles, alveolar ducts, and endpoints are alveolar sacs. It's respiratory portion of the respiratory system, and all those vesicles are alveoli. It's terminal bronchiole, respiratory bronchioles, alveolar duct, and alveolar sac. And here we can see terminal bronchiole, and here we can find respiratory bronchiole and numerous alveoli. And this region is called pulmonary acids, and part of this region which is ventilated by respiratory bronchiole is called respiratory bronchiolar unit. So it's the structural and functional unit of the respiratory portion which is ventilated by the respiratory bronchiole. And here we can see respiratory bronchioles, and they have inclusions of alveoli in their wall. And let's go to the structure of alveolus. So it's the basic unit, basic structure of the respiratory portion. It's hollow cup-shaped cavity filled with air, and where the gas exchange takes place. And let's look at the structure of the alveolus. Cells of the alveoli are called pneumocytes or alveolocytes, and there are two basic types of pneumocytes. First type, here we can see them, is called Pneumocytes or Alveolocytes type 1. It's simple squamous alveolar epithelium. Also, they are called pneumocytes of respiratory type. And also we have type 2. Here we can see them, pneumocytes or alveolocytes type 2. They are secretory cells or surfactant secreting cells. They produce special substance which is called surfactant, which lines the inner surface of the alveoli. And both types are located on the basement membrane. Here we can see basement membrane. There are epithelial structures and they have this membrane. So first type, pneumocytes type 1, it's simple squamous alveolar epithelium. You can see them here. They provide respiration, they provide gas exchange. They have very thin cytoplasm and gases they pass through the cytoplasm. Oxygen goes to the blood vessels and carbon dioxide is released into the cavity of the alveolus. Type 2, pneumocytes type 2, they are surfactant secreting cells and they produce special surfactant. It's substance which is composed of proteins of phospholipids, complex of substances, and which decrease surface tension in alveoli, which prevents their collapsing. I think you know about surface tension. If you will touch surface of water, for example, if you will put water in the glass and if you will touch, you can see that there is some surface tension of the surface of the water because molecules of the water, they have attachment to each other and they form like membrane, something like membrane on the surface. And this surface tension may cause collapsing of the alveoli because this fluid it makes collapsing of the small membranous structures, especially such as alveoli. So there is special substance which lines the inner surface, and it's produced by those cells, and surfactant is dissolved and it forms special layer inside the alveolus. Here we can see lining of the alveolus, it's surfactant. And it's also present in the bronchioles. There it's produced by the Clara cells. Here we can see alveolocytes type 1, they are squamous in shape, type 1, and type 2, they are like round or cuboid in shape, and they produce surfactants. They are present here. It's surfactant is surface active lipoprotein complex, which reduces surface tension and it prevents collapsing. It's produced by two types of cells, pneumocytes type 2 in alveoli and Clara cells in the bronchioles. Here we can see alveolocyte or pneumocyte type 2. It has lamellated bodies or osmophilic bodies, which are inclusions of surfactant, and they are released from the cell and they form inner lining of the alveoli. Surfactant, pulmonary surfactant, it lines the alveoli inside. It reduces surface tension and prevents collapse of alveoli. Here we can see this layer, and also it covers cells of type 1. Here we can see appearance of pulmonary surfactant. It has both hydrophilic and hydrophobic regions, and it lines inner surface of alveoli. It reduces surface tension and prevents collapsing of alveoli. And in some children which were born before the normal term, they haven't enough pulmonary surfactant. That's why often they have collapsed lungs, and it may cause severe complications in their newborn children. So children which were born in normal term, they have normal amount of surfactant. Also we have between first type and second type alveolocytes or pneumocytes. Also we have inside the alveoli alveolar macrophages. Here we can see macrophage or dust cell. It's present in the cavity, and some dust particles are coming here, and this macrophage eats them. It provides phagocytosis of those particles, And macrophages, they also may migrate from the alveoli to the lymph nodes of the lungs or of the mediastinum, and sometimes they carry those dust particles in the lymph nodes. Um, and those particles often are present in alveolar macrophages and in the lymph nodes of lungs in some occupation diseases, and you will study this in following years. And here we can see alveolar macrophage and its wall of the pulmonary alveolus, which is composed of type 1 and type 2 alveolocytes. And also we have here a wall of the respiratory portion of the respiratory system. And now let's look how all those functions are provided. Here we can see epithelium, it's type 1 of alveolar cells, pneumocytes type 1 respiratory type. Also we have here type 2 cells which produce pulmonary surfactant. It's basement membrane, and its surfactant which lines the inner surface of the alveolus. Here we have capillary. It's also lined with simple squamous epithelium, which is called endothelium. It has own basal membrane. And usually in the organs and the tissues under the basal membrane, we have layer of loose fibrous connective tissue. But in the lungs, we have very close localization of those structures. And two basal membranes, one of the alveolus and another of the capillary, are fused, and they form common basement membrane. Here we can see common basement membrane of two structures. And this basement membrane, it reduces thickness of the blood-air barrier, which separates air in the alveolus and blood in the capillary. Blood comes here, and this blood is venous. It's carried to the lungs by pulmonary artery. It's small route of circulation, and here blood is saturated. It receives oxygen and releases carbon dioxide. So carbon dioxide is released in the air, and oxygen goes to the erythrocytes to hemoglobin, and hemoglobin is saturated, and as a result of this, we receive arterial blood. So here we can see gas exchange in the respiratory portion of the respiratory system. It's the main function of the respiratory system. And blood-air barrier includes such structures as surfactant, the innermost layer, pneumocyte of type 1, it's cytoplasm of this pneumocyte, it's basement membrane, common for alveolus and capillary, and endothelial cell of the capillary. There are layers of the blood-air barrier. And here we can see it's capillary and it's a wall of lumen of the alveolus, and we can see alveolocyte, basement membrane, and endothelial cell of the capillary. And you can imagine how thin this layer, this wall is. And also here we can see blood-air barrier and very thin wall of this blood-air barrier. And lungs outside, they are covered by special serous membrane. It's called pleura. It's loose fibrous connective tissue and mesothelium. Mesothelium is simple squamous epithelium which covers pleura, and loose fibrous connective tissue. So there are two layers of pleura. Pleura is serous membrane, and it's very close in the structure to the pericardium and peritoneum. But note that pleura, it covers lungs outside. It's a visceral layer, and also it lines the cavity of thoracal cavity. But pleura doesn't cover bronchi. That's why they have adventitia. It doesn't cover bronchi. So pleura covers lungs, and it lines the thoracal cavity. It's serous membrane, which is covered with mesothelium, simple squamous epithelium. It's the structure of pleura. So students, that's all, and thank you for your attention.